Okay, excuse me. Welcome to this week's other video lecture where we'll start to get into spatial statistics. So the first thing we're going to be looking at in this video is looking at spatial clustering. And then the next video will be looking at spatial interpolation, aka geostats. I apologize for my voice. It's a little hoarse today, but I should be able to, so I'm going to break this lecture into two parts to make it a little shorter and save my voice a little bit. Okay, so we're going to start with spatial stats, looking at things like autocorrelation, right? And this is the question we're interested for because as spatial statisticians, we're interested in how patterns in space, right? Patterns in things, objects, populations over the Earth's surface are distributed. So spatial autocorrelation, which essentially deals with the idea that like things are nearer to more like things, right? AKA clustering. And we'll also look at things like interpolation and other approaches to do spatial stats in the second video. And just to note that some academics make a distinction between spatial analysis between point and area data. We'll not do too much of that in this lecture, but just keep that in mind. So also keep in mind that we can set these autocorrelation and spatial statistic analyses up, just like other classical statistical tests that we've done in the past, right? When we looked at the normal distribution, where we have some random variable, right? Where we can assume that we have a null hypothesis and that as our variable or a test stat gets more and more extreme, we're more likely inclined to reject that null hypothesis and go with a alternative hypothesis by looking at things like the p-value. And if the p-value is less than our alpha value, that tells us that we're going to reject the null hypothesis in favor of the alternative hypothesis. So in this first video, we're going to be looking at exclusively at spatial clustering, which is different ways, techniques that we can evaluate how points or values are clustered or are dispersed in different patterns. So there's two ways that we can evaluate how much spatial clustering we might have in a data set. So the first one we're going to look at is distance, right? So looking at the distance between one point and another, whether those 2 points are located close to one another or located far away from one another. And another one is contiguity. So contiguity has to do with what the value is in one polygon relative to what the value is in another polygon next to it. And in the next video, we'll be looking at spatial interpolation, which has more to do with how to model surfaces based on interpolation of data, basically calculating data points which are unknown between data points which are known. So in the spatial clustering part of this video, we're going to be looking at mostly at autocorrelation. So autocorrelation is going to be the main topic of this week's upcoming lab. We're going to be using Geog to assess spatial autocorrelation. And so basically we're assessing autocorrelation to answer research questions, whether a variable is clustered or dispersed over space. And we'll be looking at also different measures of what's known as global measures versus local measures. So global, there's a difference determining the spatial scale of analysis. We'll be determining the directionality, right? Positive autocorrelation versus negative autocorrelation, and the different weight matrices that we can use to perform the analysis of spatial autocorrelation of our data. And then based on the analysis or the data collected through our weight metrics, we can apply various local and global measure or statistics that can quantify how much spatial clustering we're dealing with or not. So basically we can generalize spatial clustering or autocorrelation in three main scenarios, right? Our random scenario, where basically the point locations, right? The coordinates have no rhyme or reason. They're basically randomly distributed. So we can have this in either points, like in the top here, or we can have it in grids down here in these different colors. Where we have positive autocorrelation, right, we're going to have points that are closer together than one would expect from a normal spatial distribution. And then the opposite with dispersion, aka negative autocorrelation, where we have a greater distance between our points than what we would expect if the points were distributed randomly. So we can look at spatial clustering through a research question, right, where we're trying to find out whether a certain variable or points in a location are distributed more closely to one another than you expect randomly or located further apart. So this is cluster statistics. So usually when we set up the research question in terms of spatial clustering, our null hypothesis is that there's no clustering going on. The data points that we do see, even though they might appear a bit clustered, that clustering is assumed with the null hypothesis just to be due to random error. And then we can look at, right, different types of data. We can look at clustered points versus dispersed points, clustered values, like a Z value versus dispersed value. So sometimes we're just looking at the coordinate location to see whether they're closer together or not. And sometimes we're looking at the values at each of those locations to see if the values are spatially clustered or not. Then again, we can apply both global and local scale patterns in this analysis. Talk a little bit about the difference between global and local. So global, right? Global basically has to deal with looking at your complete study frame, your study region, and determining whether your variables across your study frame are clustered, autocorrelated, or not. So with global, you're looking at whether there's autocorrelation throughout your whole study region, right? The problem is when you do a global analysis of autocorrelation, often this can mask or you can maybe not pick up on smaller spatial scales where you do have clustering going on. So sometimes looking at it too large of an area can conceal or you may not pick up on more local scale clustering going on. So this is where you have to do a local measure of autocorrelation or clustering. And there's a whole suite of different tests that you can do to look at local areas. And often when we're looking at a local analysis of autocorrelation, we're trying to see whether we have any hotspots, i.e. areas where there's like values next to like values, or cold spots, right? Where we have unlike values next to one another. So here's a map here showing local autocorrelation, right, of vandalism rates across Lincoln, Nebraska. So this can show us where we have high values next to high values of crime or vandalism in this case, shown in the red areas. And it can show us also here in the blue areas where we tend to have low crime vandalism rates next to low vandalism rates. So local measures of spatial autocorrelation can help us pinpoint some of these more local areas of either low versus low or high next to high values. So just a small list of some of the different measures of global autocorrelation that we're going to look at. Moron's eye is going to be the main one. It's going to be the main one we're going to be dealing with in next week's lab to describe autocorrelation. Some other alternative ones is Getty's G stat or Geary's C. And then some local autocorrelation measurements. A main one that we'll also be looking at in the lab is the local Moron's eye, aka LISA. The local Getty's G stat to look at hotspot clustering. So you can also think of this global versus local idea. It's kind of like what we saw with the ANOVA test. Remember that the ANOVA test, when we're looking whether there's a difference between a mean when we're analyzing 3 samples or more, the ANOVA test doesn't tell us which of those groups is different from the other ones. It only tells us if there is a difference. And once we run the ANOVA and we know that one mean or one sample is different from the others, then we have to do a post hoc test, right? Remember the Tukey test we did in the lab, which allows us to identify exactly which one of those samples is different from another one. So we can kind of use global and local assessments of clustering together, where global gives us an overall picture of where we might want to look in more closely, what areas we might want to look in more closely to follow up and do a local assessment of spatial autocorrelation. Okay, so how we actually carry out technically the assessment or measurement of our values is how we define it based on setting up what's known as a weight matrix, right? So the weight matrices, and this is something again we'll be doing in this week's lab, is basically telling a program like Geog, which we'll be using, how to look and how to analyze values around a certain location to see if they're related or not. So this is what's known as a weight matrix. So these kind of matrices, you can kind of think of them as maps or algorithms that guide our analysis, and basically tells us, the program, what points or polygons to include in the specific analysis. So in terms of the weight matrices, there's two main types that we're going to look at in this course. One is called distance. So basically just looking at points within a certain distance range around your point in question, and continuity, basically looking at what is the value of the grid or cell next to the grid or cell which is in question. So for our distance-based weights, right, again, when we're using a distance-based weight, we're basically setting a certain distance from our point in question at which the program is going to look at, right? So these might be centroids or polygons, right? So basically we set this radius, and the program looks at the values within all of those in the radius and compares them to the cell in question to determine how alike or unlike they are. The continuity is a little bit different. It's looking at how the variables, right, are different or similar to the cell in question based on, right, based on whether they're nearby or not. So we're going to look at two types of continuity weights. One is called the rook. So if this is our grid cell which is in question, the rook is going to look at the values in this cell, in this cell, in this cell, and in this cell, okay, and compare them to see how similar or unsimilar they are to the cell in question. With the queen's continuity weight, right, it's similar, but now it's going to look at also the values in these corner cells. So the queen's continuity weight matrix is going to look at more values around the cell of interest compared to the rook version. So let's get into a little bit about spatial autocorrelation now, right? And this comes back to Tobler's kind of mantra of geography, is that all places are related, but places that are closer to one another tend to be more related than more distant places. So this is the whole idea of spatial autocorrelation, where we might have values that tend to be clustered or more like items or values that are closer to one another and tend to get more dissimilar as the distance increases between the locations. So we have to keep this in mind, spatial autocorrelation clustering, because often a lot of our statistical sampling assumptions basically operate underneath the assumption that we're dealing with a randomly distributed variable. So if we have autocorrelation or clustering going on, that assumption is no longer valid, right? So again, a lot of our standard statistical tests, right, assume that either the observations or the errors themselves are independent of one another, aka there's no clustering going on, okay? But again, if we have spatial autocorrelation or correlation going on, we're going to tend to have more like values closer to one another, so that assumption doesn't necessarily apply. And so we have two cases where we do not have a randomly distributed data set. One is where we're going to have spatial autocorrelation. In other words, we're going to have clustering. We're going to have more likes, things, events, which are more similar to each other, closer together. Then the opposite of that is negative spatial autocorrelation, where we have values which are opposite, closer to one another, and we tend to have a more uniform and unnatural distribution of values compared to a random spatially distributed data set. So again, our assumption is that for a lot of spatial stat tests or sampling methods is that we're dealing with a variable which is randomly distributed, right? So if we go in here and sample, we'd imagine that the sample that I collected in that circle right there is going to be somewhat similar to any other random sample I'm going to collect within that area. But you can see that doesn't apply if we have, say, for here in the middle, where I have a very clustered data set. So in this case, if I go in and draw my circle right there, I'm going to get a very different result compared to if the areas or the values are randomly distributed. The same thing if I take those same samples from a negatively distributed or autocorrelated data set. I'm going to get very different results compared to a randomly distributed data set. So we're going to go through quickly here some measures of spatial clustering. The first one we're going to be looking at is nearest neighbor, which basically gives us an idea of whether points are located closer than to one another compared to a random data set. And then looking at spatial autocorrelation, we're going to be looking at, again, Moron's I to calculate that, and a few other ones not as well as used, Getty's G-stat, Gary's C, and then looking at some of the more local autocorrelation measures, local Moron's I, the LISA, and the local Getty's G-stat to look at hotspot clustering. So let's first look at nearest neighbor. So basically the nearest neighbor analysis, NNA, gives us an idea of the average distance between points. So it is a global measure. It doesn't give us any idea about local clustering really. And it uses point data. So we're not interested in the value at a certain location. We're just looking at the spatial distribution of certain points. So X, Y, lat, and longitude. And we can apply it to not only point data, but if we have dealing with polygons or area data, we can convert those areas to centroids. So centroids, right? It's where we have a polygon, and we basically, the centroid is the center of mass of that polygon. We can use those centroids to basically use them as point data to run a nearest neighbor analysis. So you imagine here, we've got a picture, a map of the contiguous United States with a bunch of points here. And you want to run a nearest neighbor analysis, right? It's basically measuring the distance between that point to the certain number of the nearest points around it. So the nearest neighbor run for that point right there, and you can compare that to the nearest neighbor of the distances between that point and its nearest neighbors around it. So basically the nearest neighbor index, or sometimes called the average nearest neighbor, measures the distance between each point and its nearest neighbor, right? But basically the index itself is giving us the average distance between the point in question to all the points around it. And basically what it's doing is that it's comparing the observed distance, average distance, to all the points around it, all its neighbors, compared to a hypothetical random distribution. And so if these points are closer to it compared to a hypothetical random distribution, we're going to consider that the points are spatially configured in a clustered pattern, right? They tend to be closer together than we would expect randomly. Oppositely, right, if the distance between that point in question and all of its neighbors is further than the hypothetical random distribution, we're going to consider that those points are dispersed compared to what we would expect in the hypothetical. So basically what the nearest neighbor index is giving us is the ratio of the observed average distance between the point in question and all its neighbors compared to the average distance to the point to all its neighbors of a hypothetical aerial distribution. And so basically how to calculate the average nearest neighbor is the ratio, again, of the observed average distance between the point in question and all its neighboring points to the average for what would be expected. To calculate the observed distance, basically you're taking the distance between all of the neighbors to the point in question, adding them all up, dividing by the number of points. So basically taking the average, right? That line above the D there indicates average. So this term right here is our average observed distance. And then we're comparing that in our ratio to the expected distance, which can be given by 0.5 divided by the square root of the number of points divided by the area of analysis. So basically we can combine all of these three terms together and calculate the average nearest neighbor using that formula right there. So basically the nearest neighbor index, what we're going to get back is a function of course, the average distance between the point in question and all the neighbors around it, the number of points in our data set, aka n, the size of our area of analysis, and what we're going to get back is a number that's going to be somewhere between 0 and 2.15. Okay, so how do we interpret the number that we get back for our ANN ratio? Well, 2.15, the maximum value, is a perfectly dispersed or a very dispersed data set. 1, an ANN index of 1 means that we're dealing with a randomly distributed data set, and an ANN of 0 is that it's perfectly clustered. So we can look at the results of an ANN value based on this continuum here, where again, an ANN value of 1 means that we're dealing with a randomly spatially distributed data set. If we have a value of 2.15, that means our points are very far apart than we would expect randomly. And then if we get a value closer to 0, that means we're dealing with a more clustered, spatially autocorrelated, positively data set. So if we have an RN value of 1.27 for our average nearest neighbor value, that means that we're close to random, but we're verging a little bit towards regular spacing. Oppositely, if we had a ANN of 0.8, it would be close to random, but it would be verging towards clustering. So again, the way we can visualize this with an R of, or an ANN of 2.15, that means we're dealing with a very uniform or dispersed data set. An ANN of 1 means there's no rhyme or reason to our spatial makeup. We're dealing with a random data set. And then if we have an ANN or an RN of 0, that means we're dealing with a highly clustered or spatially autocorrelated data set. And we can actually calculate the Z value for the average nearest neighbor to see whether it's what we would expect or we're dealing with a very extreme value. So just like we calculated Z values from raw data scores before, we can calculate the Z value for our average nearest neighbor by taking the observed value minus the expected value divided by the standard error, which is given by this term right there. And then we can compare the Z value for our average nearest neighbor to see where it lands on a normal distribution. So our null hypothesis again with spatial autocorrelation is that we're dealing with a randomly distributed data set. And it's only when we reject that null hypothesis is that we're either going to go with the idea that we're dealing with a dispersed data set if our Z value is very high. If we have a very low ANN, that's going to tell us, and it's on the extreme end of the tail there, that's going to tell us that we're going to reject the null hypothesis and instead consider that our data set is clustered spatially. So an example that we could apply to the average nearest neighbor algorithm or variable here is that imagine you're studying the distribution of hurricanes over the southern United States over time and space from the years 1980 to 2007. You run your nearest neighbor analysis, you calculate your ANN value, right? And you get a ANN or R value of 0.2, and your p-value, remember what the p-value is returned at 0.02, meaning that it's quite significant, and it's one-tailed. So this would be a classical hypothesis test that you could state your hypothesis and interpret the results on. So in this case, your null hypothesis would be that there's no significant clustering or dispersed spatial pattern to your data set, is that there's no significant clustering in the hurricanes in the southern United States over that time period. Your alternative hypothesis is that, yes, there is significant spatial clustering. Again, we're dealing with a one-way test here, so we're just looking at whether there's clustering or not. Imagine you set your test to be at the 95% confidence level. And then based on your results that I showed in the last slide there, with a p-value of 0.02, right? For a 95% confidence level, you're going to have an alpha level of 0.05. So this is where we have a p-value less than your alpha level. So you're going to reject your null hypothesis and go with your alternative hypothesis that there is significant spatial clustering of hurricanes across the southern United States over that time period. Okay, so that's nearest neighbor analysis. A somewhat related way that we can analyze spatial clustering is something called quadrant analysis. So quadrant analysis is basically a sampling method in which we set up a whole bunch of similarly shaped quadrants or grids, and we basically place that grid over our area of interest, and we count how many of our points of interest fall within each of those grids or quadrants. So I'm using the term grid, but your quadrant can take on any shape, triangle, hexagon, whatever you want. So basically you're setting up this quadrant and you're placing it over your area of interest, and you're counting up how many occurrences you get in each one of those quadrants. And this is a common way that we do spatial analysis and field work, right? You go out and maybe you're interested in how many nests might be in a certain area. You can go out and take a grid quadrant, you can randomly lay it down and count how many nests or plants of a certain type or species fall within each of those quadrants, right? So a couple different techniques that use commonly in fieldwork is setting up a uniform grid, and then within that uniform grid, counting the occurrences within where they occur in each of those grids there. Another common technique is random sampling, where you're randomly, by chance, selecting coordinates, setting down your quadrant, and then counting how many of the occurrences of interest fall within that quadrant. So just keep in mind, like I said before, your quadrant does not have to be a perfect grid or square. You can use any shape you want. It could be triangles, it could be honeycomb shaped, right? But the idea is the same. So we can get a bunch of different results in terms of the spatial distribution of our variable of interest. So imagine if we have, like on the left here, we set up our grids all along like this, and we count how many occurrences we have in each of these grids. We can see that we have a lot of ones, fewer twos, fewer threes, so on and so forth. So we tend to get a random pattern that we'd see down there. Now imagine again, we set up our grid quadrant analysis like this, but it gives us a very different result. We can see that we have a lot of quadrants in this case that have no values within them, indicating that we have a positively skewed frequency distribution with more quadrants with no values in them and fewer quadrants with high numbers of values within them. Now, a large consideration when setting up a quadrant analysis is selecting the size of the quadrant you're going to use. Because what happens is if you set up the size of your quadrants to be too fine or too small, you're going to end up with many quadrants with 0 points in them, right? Too small, many quadrants are going to have 0 points within them, right? So you have these very small quadrants. So you can see a lot of them are going to have 0 values or 0 observations within them. And therefore, you're going to have most likely a highly positively skewed frequency distribution. But if you, on the other hand, if you set them up too big, you're going to have many quadrants that your quadrants are going to have basically all the same number of values within them. Okay, so if your quadrant is too big, you're going to get a uniform measurement of your spatial distribution. So kind of the middle and the optimal size, right, is capturing the spatial distribution of the variable of interest that you're after. So one commonly used way to calculate what size you should go with your quadrant is square root of 2 multiplied by the area, your studied area, by the total number of points that you're observed within the study area. So again, your quadrant can be any shape, circle or polygon. And the way that you usually want to analyze the variance is to look at the relative variance, like the coefficient of variation of the points between the quadrants themselves. And so when we use a grid, the quadrant analysis is basically a type of global measurement of correlation. And then if we move the quadrant around within the area, it can be used as a local measurement. So here's an example of a local measurement where our quadrant is a circle and we're moving it around our map of the United States along the west and south coast here and counting each time we move it, how many values fall in each of these instances of our quadrant. That would be an example of a local measure. And again, we're after what type of spatial distribution is our observations in. Are they clustered? Are they relatively random? Or are they uniform in nature? So if we do get a certain random distribution to our data set, we can compare that to what we would expect it. Remember when we calculated Poisson distributions to see whether our observed distribution is random or not, right? So again, if we have a randomly distributed data set like we have in here, we expect a close correspondence between the observations, which is in black bars here, compared to the expected Poisson distribution, which are in the red bars. If we have a clustered data set, right, we can see we get a big difference in the number of observed grids with no values in them compared to what we would expect. So we get a big difference or discrepancy between the observed number of points within each quadrant compared to the expected Poisson distribution in that area of our histogram. And then with if a dispersed spatial variable, the opposite is going to be true. We have more of a difference in the quadrants with a high number of points within them. So we can use quadrant analysis to look at the distribution of cell frequencies, and then we can basically get an idea of the point pattern arrangement from that. Right, so remember in nearest neighbor, we were looking at the average spacing between a point of interest to its nearest neighbors around it. In quadrant analysis, we're looking at the variability and the number of points in the quadrants around it. So slightly different here. An important thing to keep in mind here is that we're going to look at the variance, but we're not going to be looking at absolute variance like standard deviation. Instead, we have to look at the relative variability of the number of points within the quadrants around it. So what do I mean by this? So imagine these three examples here. We have a random quadrant analysis at the top, uniform dispersed in the middle, and clustered at the bottom. So we can use something like the coefficient of variation, which you recall is the standard deviation divided by the mean of the data set, to assess the amount of variation in the point dispersion between our quadrants. So if we have a uniform distribution, our variance is going to be 0 or very close to 0. So when we have the variance, right, standard deviation close to 0 divided by our mean, so we're going to have something close to 0. So for a very uniform or dispersed quadrant analysis, right, we would expect a coefficient variance very close to 0. On the other hand, if we're dealing with a randomly distributed data set, quadrant analysis, the variance analysis of it is going to give us a coefficient of variation somewhere around 1, right? Because we have some variance, right, difference between the number of values within each of our quadrants. And then if we're dealing with a highly clustered data set, we're going to expect a coefficient of variation above 1 because there's a big variance or difference in the number of points within our points relative to our quadrants relative to our mean. So to calculate the variance to mean ratio, right, we basically take the variance of the cell frequencies in the top here divided by the mean cell frequency themselves. Then we can apply, right, we can take the number of points and the number of cells here, and we can plug those into this equation here. Don't worry about the equation, but this is basically how we get at calculating the variance. It's like the coefficient of variation. So again, when we're dealing with very dispersed or regular distributed set of points, the cell frequency is going to be very similar, so our variance is going to be relatively low. If we're dealing with a highly clustered, the variance is going to be high, so we're going to get a relatively large variance. And then if we're dealing with a relatively randomly dispersed data set, we're going to get a VAR somewhere around 1. So an example here, imagine you're interested in determining whether the spatial distribution of tornado touchdowns in Illinois is random or not. And to do this assessment, right, you have 450 points, tornado touchdowns, points, and you have 63 grids or quadrants in this case. So basically what you'd be doing is comparing the variation of the number of cells or number of touchdowns points within each of these quadrants to see how much they vary or not. So you would take your data, right, kind of like with the Poisson distributions before, where you would have the number of tornadoes, the number of times that number of tornadoes was observed, right? So for example, 3 tornadoes was observed 7 times, 13 tornadoes was observed 0 times, 14 tornadoes observed 4 times. So that's quadrant analysis and nearest neighbor analysis. I'll move on to something called Ripley's K. So Ripley's K is a bit different because it shows us how the degree of clustering can change with changing the distance at which we're analyzing around our point of interest. Right, so we use Ripley's K because often we're interested in getting the right scale or the right radius of analysis to do our spatial analysis investigation. And in this case, we're applying Ripley's K to point data, and it's a local measurement that, again, is looking for the appropriate scale in which to do our analysis on. So what am I talking about here? Well, with Ripley's K, you can imagine the middle point here, that's our center, and further we go out from it, that distance is our radius. So imagine we're interested in analyzing the spatial distribution of the points away from our center point. Basically, this is what the Ripley's K gives us. So as we start to go out, as our radius shows us down here, as we start to move away from the center of this bullseye here, in this case, right, so this distribution right here, if our line follows this pathway right here, that means we have a randomly distributed data set, all along different distances away from that middle point. This purple line is showing us exactly what our observed clustering or dispersion is. So you can see here, because the purple line is well above and outside of the blue line here, at shorter radiuses or shorter distances away from our point in question, the center here, the spatial distribution of our data tends to be clustered. But you can see as we start to get further and further away, as our radius starts to get larger and longer away, so imagine out here somewhere, we can see that we cross over from a clustered spatial distribution to a more dispersed spatial distribution. So you can see by using this Ripley's K, it shows us how our clustering or our spatial arrangement changes with the distance in which we analyze away from our point of interest. So an example here, imagine we have a pine tree here and different types shown in the green. So all these green dots are pine trees, red dots are different types of pine trees. And we're interested in seeing what the type of spatial distribution is as we move away from our center pine tree away. In this case, right, we get a similar kind of observation as I showed you in the last one. At close distances or small radiuses, we tend to have clustering. But as we start to move further and further away, as our radius gets longer, our spatial dispersion moves from a clustered pattern to more of a dispersed or uniformly distributed pattern. So that's how that Ripley's K can be used. Okay, another method used to analyze spatial distribution is what's known as joint count. So joint count basically usually takes grid data, and it's basically analyzing for one cell. So imagine this is my cell here in the top left corner. It's analyzing the value of the cells at which it's joined up to, okay? So it's basically analyzing, right, what the value is in one grid compared to the other values that might be around it. So for example, for this cell right here, you'd be looking at what the value is in that cell, in that cell, in that cell, and in that cell. So kind of like a rook analysis. So you can look at the values, right? And you can convert points, right? The number of points found within a quadrant, and convert those into a grid. And then from that, you can run your joint analysis. So imagine we're looking at a 6x6 grid, okay? So if we had a 6x6 grid where we have a very highly clustered data set, so you can see here we have most of our ones together, most of our zeros together. So again, highly positive spatial autocorrelation. In this case, we have 27 one-to-one joins, right? One next to one next to one. We have 27 zero-to-zero joins, right? A zero next to a zero, zero next to a zero, and then 6 0-to-1, right? There's our 6 0-to-1 joins along the middle here. This is much different from what we'd expect a randomly distributed data set to be. Now let's look at a very dispersed data set, right? In this case, we have 60 joins, which are 1-to-zeros, right? 1-to-zeros, 1-to-zeros, basically negatively autocorrelated. So in this case, we have a lot more 0-to-1 joins than we would expect just by a random chance. Scenario C shows us a relatively random data set. So if we do a joint analysis on a random data set, in this case, we get around 35 either 1-to-0 or 0-to-1 joins, right? 1-to-0, 1-to-0, 0-to-1. We're getting about 35 of those compared to 30 would be expected for a perfectly randomly distributed data set. So not perfect, but close. We get 13 1-to-1 joins, so 1-to-1, 1-to-1, and 12 0-to-0 joins, 0-to-0, 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 compared to 15 each. So you can see by comparing what the expected join count is going to be compared to what you observed, that can tell you whether you're dealing with a clustered or more regular dispersed data set compared to random. So just some other things that you can look at in terms of applying the joint count to spatial distributions. Now let's move on to Moran's I, because this is the main stat that we're going to be using in the lab and looking at it in more detail in the later lectures in this class, right? So with Moran's I, it's like the join count, but instead of just using grid data, it's using continuous data instead of categorical, right? With the join count, we're like in the example previous, we're looking at binary, right? Ones and zeros. But Moran's I does the same analysis, but it's using continuous data. So this could be anything, temperature, voting intentions, what have you. So it measures the clustering of a magnitude of value over space. It can be performed both on point data and polygon data. So it can also be applied to give us a global measure of autocorrelation, so for your entire study area. And then it can also be applied using the local version to identify hotspots, where you might have high values next to high values, low values next to low values, high values next to low values, and low value next to high value clusters. So the main metric that comes out of a Moran's I analysis is the Moran's I slope. So we all looked at correlation, where we calculated the R value, the correlation coefficient for a XY scatter plot. The Moran's I analysis returns something similar, but instead of an R correlation coefficient, it returns the Moran's I. So the Moran's I tells us the nature of the autocorrelation, whether there is autocorrelation or not. So you can see in this plot here, it split the plot up into 4 squares. Up in the top left, this is where we would have, there are points fall into this quadrant. We would have low values next to high values. In this quadrant here, we'd have high values next to high values. Down here, we'd have high values next to low values. And down in the bottom left here, we'd have low values next to low values. So whenever you have a positive you draw the line through your data set, such as in this one, where you have a positive slope or a positive Moran's I, which in this case is 0.3990, that indicates that most of your points are falling within the low quadrant or the high quadrant, indicating that you're dealing with a positively autocorrelated or clustered data set. So the way to interpret the Moran's I is that if you get a Moran's I of 1, that's a perfectly positively autocorrelated data set. 0, right, when you have no slope, like a flat line, it'd be your data is randomly distributed. And then if you have a -1 or a negative value to a Moran's I, that means you're dealing with a more dispersed or regularly spaced data set. But remember that the Moran's I is not the only thing that you're using to determine whether your data set is autocorrelated significantly or not. Like other statistical tests, we also have to look at the p-value to see if it's significant or not. So again, just to reiterate, the way to interpret the slope or the value of the Moran's I is that if your Moran's I is positive, right, so you have a positive slope, that means that most of your data values are falling within the low quadrant and the high quadrant, meaning that you have, when you have a low value, it tends to be located next to a low value. When you have a high value, it tends to be located next to a high value. In other words, clustering, aka positive autocorrelation. The opposite, right? If you have a negative Moran's I, a negative slope, right? That means that more of your observed values, right, tend to be, when they have a low value, they're located next to a cell or another area which has a high value. And when you have a high value, it tends to be located next to a lower value. So negative autocorrelation or uniform distribution. You can think of Moran's I kind of like the correlation coefficient R that we looked at in a couple lectures past. So the R, of course, looks at the association between two variables. So imagine education versus income. You can have a positive R, which tells us that in general, as education increases, so too does people's income. A negative 0.71 or a negative R value is the inverse relationship, right? In this case, as price goes up, quantity of that entity goes down. So we can interpret the Moran's I kind of similar, but with the Moran's I, we're looking at how the variable changes over space. So if we have a Moran's I of 0.71, we might be looking at something like comparing crime rates versus the crime rate in the nearby area. So if you have a positive Moran's I, that means that basically if you have a crime rate that's high in one area, the areas around it will also tend to have higher crime rates. Say you got a Moran's I value of minus 0.71. This might be a case where you have a variable grocery store density, so how many grocery stores you have in an area compared to the density of the grocery stores nearby. They have a negative relationship, meaning that you don't want to have too many grocery stores located close to one another, right? Because there'd be an oversupply. So in this case, they tend to be spread out or more uniform than you would expect naturally. Right, so again, the Moran's I is looking at how the clustering of a magnitude of a value occurs over space. Again, we can apply it to points or polygons. It can give us a global measure or a local measure of spatial autocorrelation by looking at the clusters themselves. Okay, so how is the Moran's I calculated? So there's this kind of long drawn out equation right here, where n is the number of observations that we're looking at, x bar is the mean of a certain variable, xi is the value of that variable at a particular location, and then xj is the value of the variable at the location located right next to xi. So xj is located next to xi, and they're related to one another, xj and xi, by the weight indexing matrix. So again, basically Moran's I, we can think of it along with a continuum going from -1, which is perfectly dispersed or uniform distribution. Moran's I of 0, basically telling us that we're dealing with a randomly spatially distributed variable. And then a Moran's I of 1 means that we're dealing with a highly clustered spatial variable. So again, looking at this equation again, W gives us the number of connections in the matrix. So the W for point I and point J can be assessed by whether the cells are located next to one another, how far they are located to one another, or you can use a combination of both. And so basically this part of the equation here is computing the difference between the value at the next location compared to the mean. And then it's multiplying that by the value at the cell in question minus the mean. So you're taking this cross product of the value at point I to the mean, multiplying by the difference between the value at point J to the mean. This is what's known as the cross product. And that's the main function in which Moran's I is calculated. So again, Moran's I, the result's going to be somewhere between -1 and +1. When our autocorrelation is high, right, the coefficient is high, doesn't necessarily mean it's significant. We have to look at the p-value for that. So when we have a high positive value, it means that we're having lots of clustering going on. 0 means we're having a relatively random data set. And then with Moran's I, we can also calculate the z value, or z value, I should say, for the Moran's I to see how significant or unusual it is by comparing it to a normal distribution. So we can take the Moran's I value that we get, we can subtract from it the expected Moran's I value divided by the standard error of the Moran's I value to see how rare of obtaining that Moran's I is. And that'll help us determine whether we're going to reject or not reject the null hypothesis. So again, we can look at the Moran's I in terms of a normal distribution, where our null hypothesis is that our Moran's I is going to be either 0 or somewhere very close to 0. And as that Moran's I gets more extreme in value away from that 0, we're more likely to reject the null hypothesis that we're dealing with a randomly distributed data set and assume that we're dealing with a non-randomly distributed data set. Right? So if we get a Moran's I way out here, that means that there's a less than a 5% chance that the Moran's I that we got was just due to random chance. There's likely something going on in terms of the spatial distribution of our data set. So again, right, we can look at both sides in terms of, right, for the Moran's I, our null hypothesis is that our data variable is randomly distributed in nature, right? And any non-random pattern that we're seeing is just due to random sampling. It's only when we get very significant values of Moran's I, if they're very high values, positive values, we're going to reject the null hypothesis and consider that we're dealing with a highly clustered data set. The opposite, if we get a negative Moran's I, which is very extreme on the left tail, we're going to reject the null hypothesis and consider that our data set is more uniform or dispersed than we expect randomly. So just another resource if you want to check out a couple more examples of Moran's I in Excel right there. So we run a Moran's I test like we do in the lab. So something we want to do, say when we're using the program Geog, is to analyze our data using classification, maybe standard deviation or a natural breaks, to see how values vary with spatial distribution across our study area. So that helps us to identify areas, once we do run the Moran's I, where we might expect to be some clustering or dispersion going on. So again, the main metric that we get from the Moran's I is the I, or the slope value itself. And we can interpret this, right? Positive values indicating clustering, negative values indicating more regular or dispersed spatial correlation. But even though if we have a relatively high Moran's I either way, we have to look at the p-value to determine whether that correlation or autocorrelation is significant enough, is significant or not. So again, we're looking at, right, say if we get a Moran's eye like this, 0.486, indicating that we have pretty strong clustering going on, we got to compare that to what we would expect for a random distribution and do a test to see whether what we actually observed is significantly different from what we would expect randomly. So in terms of doing a local Moran's eye test, right, our LISA, we can look at a certain part of the data, right? So in this case, in Geog, I've dragged over top of the data in the high quadrant there. And that's basically telling me in my study area where I have positively spatially autocorrelated areas, high next to high, low next to low. And then I can perform the LISA in Geog. Again, that'll show me where I have low next to low, cold spots, high next to high, hot spots. And I can also determine the significance values, the P values, determine whether and how significant those hot spot and cold spot clusterings are. So, right, some of the outputs that you'll get from Geog, right, again, it'll tell you your P values, a little blurry there to see, but it tells you the significance of those certain hotspot and cold spot results from your localized Moran's eye test. So P values and telling you how significant each of those types of clustering that you get from your local eye test, Moran's eye test is. So with the with the Moran's eye, there's two main approaches in determining significance. One is, I mentioned already, is looking at normality, right? How expected are the results of your Moran's eye test compared to, say, like a normal distribution of the expected values of that Moran's eye? Another one that we're going to see, and we haven't looked at yet in Geog, is randomization, where basically what Geog does in the Moran's eye test is that it calculates a whole bunch of permutations using what's known as a Monte Carlo simulation. So all these permutations are basically different possible combinations of the spatial arrangement of your variable. And basically what it does is it compares all of those random configurations of the variable compared to your observations to see how likely or unlikely your particular spatial arrangement is. So again, basically what's going on is that you're taking your spatial autocorrelation and it's comparing your data to a theoretical random data set in order to get a p-value. So in Geog, it's comparing all of these permutations, so it calculates something called a pseudo p-value, because what it's comparing it to are basically kind of modeled or simulated values. They're not really values they're comparing it to, but they're computed values. And so it uses this to develop what's known as that pseudo p-value, which you can use to determine whether you're going to retain the null hypothesis or reject the null hypothesis. So essentially what the permutations are doing, so imagine you have an observed distribution of points across X and Y coordinates right there. It's comparing your observed distribution to a spatially random distribution. So this is just one possible spatially random distribution, but with Geog, you're going to be doing 500, maybe 1,000 different permutations. So you have a greater sample of randomly distributions to compare your observations to. And based on that comparison, it's going to calculate that p-value. And again, your p-value is basically telling you the probability that you would see your observed spatial distribution simply by chance. So the lower the p-value, the more likely that you have significant autocorrelation going on, whether it be positive or negative. Again, the p-values are dependent on the number of permutations that you're going to be calculating. So in Geog itself, so for example, imagine this is your result. This is your scatter plot of your values. you get a Moran's eye with a very low slope, indicating, right, and there's our line right there, very low slope, so indicating that there's probably not a lot of autocorrelation on. We're probably dealing with a randomly distributed variable. And then what you're going to do is calculate and run the permutations, and that's going to give you your pseudo p-value. In this case, we get a p-value, pseudo p-value of 0.368. So again, remember the p-value tells us the probability that we're going to commit a false positive or type 1 error. So that yellow line right there is basically showing you where your observed distribution would fit into a expected distribution. So you can see that it fits in pretty much somewhere in the middle, right? So that's telling us that, yeah, what we got in this observed distribution is basically normal. We cannot discard the null hypothesis that our distribution is randomly distributed. Opposite case here, right? Now the yellow line of our observations, our spatial distribution, is way out here, way outside of the expected distribution, giving us a very low p-value. So basically telling us that we have a 0.2% that the spatial distribution that we observed happened just by chance. And again, this is for a test run with 409 permutations. So 499 different randomly obtained distributions compared to your observed distribution gives us our pseudo p-value. Okay, so we'll wrap up, look at a couple more measurements of autocorrelation. Now, one problem with Moran's eye is that it's based on global averages, and therefore it's easily biased with outliers. Remember how way back in our early discussions about stats, how the average can be more biased by an outlier compared to the median? Well, Moran's eye is very susceptible to change based on just a couple of extreme outliers. So Gary Sees deals with this because basically, not to get into it, is that the equation that it uses to calculate the C-stat does not rely on that cross product that Moran's eye does. So this is the equation to calculate Gary's C. Again, don't get too worried or wrapped up in the equations themselves. Just know that the Gary's C is sometimes used because it's less susceptible to be affected by extreme outliers. One disadvantage to Gary's C is that it's often harder to interpret than Moran's eye. One good thing about Moran's eye is that you have a positive value, you have positive autocorrelation. You have negative Moran's eye value, you have negative autocorrelation. The C-stat is a little less straightforward, but a value of Gary's C that's between 0 and 2, right? So the value is going to be somewhere between 0 and 2, where 1 indicates that you have no spatial autocorrelation. Values lower than 1 means that you have positive spatial autocorrelation, clustering going on, and then Gary's C is higher than 1 indicate that you have more regularly spaced spatial distribution. And then we have the Getty's G, which you can apply to points or polygons, right? This is a global measure that calculates where we have high or low clustering. So basically where we have positive autocorrelation going on and what type of clustering we have going on. And we can apply the same Z-score and p-value to judge whether the Getty's G that we're dealing with or whether we get back is significant or not. So this is just showing you the slide of how the G-stat is calculated. Again, using the same set of values, right? Our weight matrix, the value of some variable at location I, at location J. You can also calculate the Z value for your G to see how expected or unexpected it can be across a distribution. Then we have the Getty's ORG G, which is basically the local version of our G. And this is used for hotspot analysis, right? Looking at more local areas of autocorrelation. So of course, a high Z-score and a small p-value indicates that we have a significant spatial clustering of high values. A negative z-score and a small p-value indicates that we have a significant clustering of low values or cold spots. So basically, the higher or lower the z-score, the more intense the clustering, and then we use the p-value to determine the significance of that clustering. So if we run a local Moran's I to determine hot spots and low spots where we have localized clustering going on, and we compare that to, say, a Getty's or G, we can see that we get generally similar results back. Some small differences, right? We can see in general terms, we get similar results back from those local measurements. Then the last one we'll talk about here is called the local Getty's Geog G stat, right? So again, a local measurement of auto clustering, where the G will be high where the high value is cluster, and the G value will be low where the low value is cluster. And we compare the G to an expected value to see how random or non-random our spatial distribution is. So let's compare and look at data for crime in Columbus, Ohio from 1995. So we can do the local cluster analysis, say using a LISA like we'll be doing in the lab. So we can identify areas where we have hot spots, cold spots, so low values next to low values, high values next to high values. The Getty's G, we can get different results based on the range or distance at which we do our analysis. So this is a Getty's with a 0.5 distance. So you can see we have hotspot clustering as we get with the LISA. But as we increase the distance, right, we can get very different results. So that's one thing to keep in mind when looking at the Getty's G. So results with the Getty's G can vary significantly based on the distance or how far you're looking around in your area. So again, just to review and wrap up for today, what type of test you're going to look at to determine your autocorrelation. First thing is the type of question that you're asking, right? Are you wanting to see whether you're looking for clustering over your total study area, or if you want to look at more specific areas? What is your data? What's the format it comes in? Is it points, right? Or is it areas? And then what are you measuring? Is it just X, Y locations, like you saw with the nearest neighbor analysis, or you have some variable or value associated with each of those coordinates? Okay, so we'll wrap up spatial clustering for today. And then next week, we'll get into the second part of spatial stats, which will be spatial interpolation. Have a good weekend.